Hello there and welcome back to another CPD talk. My name's Jasmine and I am a senior biomedical scientist in blood transfusion. Today we're going to be continuing on with our discussion about blood donations. This is part 2, so if you do want to start from the beginning, please find part 1 linked in the description below. So, how do we turn whole blood into separate blood components? We're going to go through whole blood processing, information about red blood cells, platelets, plasma, and then non-standard components. So today is very much about what you get out of the donation, whereas the previous presentation was about the donation process itself. So let's get started. Components are prepared from whole blood via differential centrifugation. Using centrifugation to separate out the different parts of the blood based on their density. Red blood cells are the densest components, so will be at the bottom of the bag, followed by white blood cells, platelets, and then plasma. So when you spin, so centrifugation is to do with spinning the blood bag, you then end up with this separation occurring. So the collection bags will be set up in 2 different types of configuration depending on the desired separation method. So when they do get whole bloods through the lab, they'll have a look at the donor's history, and the whole blood will be selected to do different things depending on the history and what's required at that time, as well as the blood group. So the Buffy Coat collection set, also referred to as B1, is used in the production of red blood cells, plasma, and platelet components, whereas the whole blood filtration set, also referred to as B2, is used in the production of red blood cells and plasma components, including cryoprecipitate. So the main difference is that in B1 you get platelets and in B2 you don't. I have then given us some pictures of the 2 methods. So if we have a look at the Buffy Coat one, so this is the one where you get platelets, you can see that the whole blood is that first image. It's cooled, but more towards the room temperature, so between 18 and 24 degrees. It's centrifuge. And then, as I said before, you can see the separation here. You've got red blood cells at the bottom, you've got the platelets and white blood cells, and then you've got the plasma. And then these things are extracted, so they're separated off. So you have red blood cells, Buffy Coat, and plasma. The Buffy Coat then will be processed into pooled platelets, which we'll go into in a minute. The red blood cells have an additive solution added to resuspend them, and then they will go through filtration, also known as leukoreduction, to remove any remaining white blood cells that might be present to give you that finalized red blood cell unit. Whereas in B2, you again start off with the whole blood. It's cooled to between 1 and 6 degrees, so it's cooled a lot lower than with the Buffy Coat method, and the filtration that I just mentioned is actually done here. So this filtration will remove not only any white cells remaining, but it will remove the platelets as well, which is why you won't get any platelets out of this method. Again, it goes through the centrifugation and then you get the extraction, but you can see in this picture there's no Buffy Coat, so there's no platelets or white blood cells. So looking at the red blood cells specifically, whole blood collected in CPD anticoagulant is processed by either the B1 or B2 method. In the B1 method, the whole blood is centrifuged to separate out red blood cells from platelets and plasma. The red blood cells are then leukoreduced by filtration. Whereas in the B2 method, the whole blood is first filtered to remove the platelets and leukocytes, then centrifuged to separate the red blood cells from the plasma. In both the B1 and 2 methods, the red blood cells are then mixed with an additive solution. This is normally saline, adenine, glucose, mannitol, or SAG-M, and then labelled as a red blood cell unit. All red blood cells prepared by NHSBT will have undergone this leukoreduction, and the average volume of a red blood cell unit issued by the NHSBT is 270 mils plus or minus 50 mils. The hemoglobin of this unit should be greater than 40, with a hematocrit of approximately 0.55 to 0.75. Further modifications of red blood cell components will be discussed later. One of the samples taken with a donation is tested for various antigens and for a number of infectious diseases. In addition, all donations undergo the ABO and RHD typing and are labelled accordingly. KEL typing is performed on every donor twice, and when negative, this result is printed on the label, and a proportion of donors are also phenotyped for additional red cell antigens corresponding to commonly clinically significant antibodies. I've given a couple next. Negative results for any of these antigens appear on the label, and the complete set of results, including positive and negative antigen tests, appear in the barcode label. So, having a look at all of this information, the last paragraph that I've gone through where I'm talking about KEL and phenotyping, if you haven't seen any of the talks where I have explained blood grouping, this is not going to make much sense to you. But in general, KEL, the ABO you've probably heard of, and the RHD you've probably heard being called rhesus, but it is RHD type. So for example, you can have someone who is O RHD positive or A RHD negative. Those are the main blood groups that you'll be aware of and that you might know your own of. As well as that, everyone's red blood cells has many other antigens on them, and we are looking at the antigens that can cause an immune response as a result of sensitization. So KEL is one that we are more careful over because it's very easy to sensitize someone. And the way you sensitize someone is if you get some donor red blood cells that have that antigen present on them and you transfuse them to someone who doesn't have that antigen on their red blood cells, then there is a small chance that you may create an antibody to that antigen. We're most concerned when this happens with patients who could get pregnant or are pregnant, because that can lead to issues with the fetus, or patients who are multiply transfused, because once you have one antibody, your system is more likely to create further antibodies, and then it becomes more complicated for us to select blood. So when we say KEL typing is performed on every donor twice, and when negative it's printed on the label, in the UK it's pretty much standard that if you have a female of childbearing age or under childbearing age, then you would give them a KEL negative unit to prevent this sensitization from happening. So that's why KEL is specifically mentioned here along with the ABO and RHD. So this is a picture of a red cell blood unit. It has a lot of information on it, but if we just have a look, we're going to start near where it says the volume of the unit. So you've got the barcode next to that tells you what product it is. So in this, it will tell you that it's a red blood cell. If you were to scan it in, that's what the computer will recognize it as. Above that is the donation number of that unit. So that's a unique number that is given, and that's how we identify that unit and what happens to it. If we then go to the right hand side of that, those barcodes have the information about the blood group, so the ABO and RH status, which is then printed in those big letters for us to notice below that. Then the next barcode underneath that has the expiry date information, and then in the additional information section, that's where any of the antigen information is. And anything that's printed on there, and the majority of things that are printed on there are telling us that it's not present. So if we have a look at this patient's one, for example, you can see underneath you've got the DCE, little c, little e bits there. It will have a negative or a positive telling you whether that's present. Underneath that, it says that it's HBS neg and CMV neg. And the bit where it says neg next to it, these are then when we're looking more at the antigens. So all units now have to be HEV neg. So that's hepatitis E. That's a standard in the UK. The K neg, that's to do with the KEL, as I was saying about how we have to give patients of women of childbearing age KEL negative blood. Then you've also got FYB, JKB and CW, which are other types of antigens that you can get antibodies. So if we have a patient who needs a transfusion, their blood group is O negative and they have an antibody against FYB, then we would probably select this unit as a possibility for transfusion. It's really important all of this information is contained within the barcodes as much as possible, because it reduces the risk of human error. Whenever a blood unit is sent to a hospital, these units are booked in, and it's a legal requirement for us to have all of the information from the point of it being taken from the donor to the point of it being given to the recipient. So we want to eliminate as many chances for human error as possible. So having those barcodes where we just scan this into our system and it downloads all of that information makes it safer for the patient on the other end, and it means that our records are more accurate. If we move on to platelets, as we previously covered, there are two types of platelet preparation, the pooled and the apheresis. Pooled platelets are prepared from whole blood collected into a Buffy Coat collection set, so that's the B1, with CPD anticoagulant. The whole blood donations are cooled to room temperature after collection. After transportation to the production site, the blood components are separated by centrifugation. The top layer, containing the plasma, and the bottom layer, containing the red cells, are extracted. The Buffy Coat layer that contains the platelets and white cells, though, from between 3 and 5 donations of the same blood group, along with plasma from one of the same 4 donations, and it has to be a male donor, I can talk about that later, are pooled together and processed further, including leukocyte reduction by filtration, in order to produce one pooled platelet unit or dose. This pool is labelled as Rh negative only when all the donor units within the pooled component are Rh negative, and the pooled platelet units are produced within 28 hours of collection and have a unique pool identifier number. They have an average volume of 317 ml plus or minus 16 ml and a typical platelet count of 339 plus or minus 44 10 to the 9 per unit. Their shelf life is 7 days from time of collection. The typical unit of pooled platelets have an average residual leukocyte count of 0.04 and may also contain trace amounts of red blood cells. So that was a lot of information. A couple of things to note. When you have the Buffy Coat, obviously that's going to be the smallest layer that you're getting out of this whole blood donation. So to make it into a proper pool of platelets, we're taking it from between 3 and 5 donors. Where possible, they will all be the same ABO and RH group. If the RH group cannot be kept the same, then the platelets will be known as RH positive, because it's safe for RH positive patients to have RH negative blood, but the other way around can cause issues in some circumstances. The fact that you have pooled between 3 and 5 donations means that if you get a transfusion of platelets, you're actually being exposed to between 3 and 5 donors. And there's a lot of debate over the risks associated with this, because obviously where possible, if you can reduce the amount of donors that you're being exposed to, you're reducing the risks involved. However, one of the arguments with pooled products is that the amount that you're being exposed of from all of those donors is a lot less, and therefore there's a lot less chance that you're actually going to have any issues as resulting from that. The other thing that's important to note from this is that they can contain small trace amounts of red blood cells. This is important because this is why transfusion of platelets will affect your sample validity, because you can still create antibodies if there's been enough red blood cells in that platelet pack that you are sensitized to them. It's very, very rare and very unlikely, but there is that possibility. And that is also why we will do our utmost to try and give patients ABO matched platelets and not just select suitable units unless it is an emergency situation. Apheresis platelets, however, are collected and prepared by the apheresis machines that separate and extract platelets. They're suspended in the donor's plasma, and a typical unit of apheresis platelets issued contains about 333 plus or minus 34 to 10 to the 9 platelets, with a mean residual leukocyte count of 0.09 and an average volume of 223 plus or minus 9 mils. So you can see here the actual values are very similar. You tend to get less platelets volume when you're being transfused an apheresis unit. Obviously, you're only being exposed to one donor, but you will be being exposed to more from that one donor. And there's a slightly higher leukocyte count in the donations taken from apheresis platelets. So there's a lot of debate between which one is better, whether pooled or apheresis platelets are better. In general, you'll just follow your trust's/hospital's policy. It was a standard up until recently that all neonate/pediatric patients would be given apheresis platelets to try and reduce donor exposure, but after several research have not found any damage in having the pooled platelets. This is no longer a requirement. It is just down to your trust policy. If we have a look here, this is going a bit more in detail of the Buffy Coat method that we saw before. So you can see you've got the you got 4 Buffy Coat donors, so between the 3 and 5, and then the donor 1 you've also got the plasma unit from. You can see that there are some red cells in each of those donor units that have just been extracted and are still in there. These are then all combined into that one bag. You have further centrifugation and then extraction of any remaining red blood cells, and then the filtration of the white blood cells. So then you should be left with just the platelets and the plasma. However, occasionally you do have the tiniest amount of red blood cells left in that unit. We're going to move on to plasma, which is the final big hitter of the blood components. So again, blood donors are screened for the presence of antibodies to red cell antigens, so it's kind of the opposite this way. And if a clinically significant antibody is identified, then the plasma is discarded. So this is just so that you don't end up with a reaction of the donor plasma against your red cells. The fresh frozen plasma is prepared from whole blood collected in CPD. That is red blood cell reduced by centrifugation. FFP produced by the B1 method is both leukoreduced and platelet reduced by centrifugation, but not leukoreduced by filtration. Whereas FFP produced by B2 method is both platelet reduced and leukoreduced by filtration. The extracted plasma is frozen within 24 hours of collection and labelled as fresh frozen plasma unit. The processing steps inherently reduce the number of leukocytes to a residual leukocyte level that averages less than 5 to the 10 to the 6 per unit. And because individual unit leukocyte levels can be highly variable, FFP components are not labelled as leukoreduced. Apheresis fresh frozen plasma is collected by an automated inline process using apheresis machines that separate and extract plasma from cellular whole blood components by centrifugation. Plasma collected by apheresis is frozen within 8 hours of collection and labelled as an AFFP unit. So you've then got cryoprecipitate, which is prepared from slowly thawed FFP that is centrifuged to separate the insoluble cryoprecipitate from the plasma. The plasma is removed and the insoluble component is refrozen and labelled as cryoprecipitate. Typically 1 unit of cryoprecipitate, 10 plus 2 mils, plus/minus 2 mils, is obtained from 1 unit of FFP. Each cryoprecipitate contains a mean of 285 milligrams plus or minus 88 of fibrinogen. The average volume of FFP and AFFP components issued by the NHSBT and their coagulation factors content are on the next slide. The coagulation factors 5 and 8 are liable coagulation factors and are not stable in plasma stored for a prolonged period at 1 to 6 degrees. Therefore, plasma is stored in the frozen state I'm going to have a look now at some non-standard components. So as well as the routine components discussed, there are several non-standard components where extra steps are taken to make them suitable for different patient groups. These include frozen and deglycerolized red blood cells, irradiated components, washed components, and granulocytes. So frozen and deglycerolized red blood cells. So there are some red blood cells antigens that are known as high prevalence or high incidence antigens. I mentioned these before. This means that greater than 90% of the population have these on their red blood cells. Patients who do not have one of these antigens on their red blood cells are seen as having a rare blood type. In some of these cases, the NHSBT will freeze donated blood so that it can be thawed in an emergency for a patient who has an antibody directed against the antigen. Cryopreservation by freezing slows or suspends most metabolic functions and will therefore limit cellular degradation. However, the method of freezing must be exact so as to prevent the red blood cells from lysing. So anyone who's done any science will know that red blood cells, the way that they are optimized for oxygen crossing backwards and forwards over their membrane, they're therefore very, very sensitive to changes in temperature and pressure in the charge around them. So if you were to just stick a bag of blood in your freezer, you probably lyse all the red blood cells, which then would become dangerous if we transfused that, because it's the internal parts of the red blood cells which can cause issues for your spleen and other organs as they're having to clear it out. So to minimise the effects of this on red blood cells, a cryoprotective agent, normally glycerol, is added to the red blood cells. There are two methods to achieve this: the low glycerol rapid cooling method and the high glycerol slow cooling method. In the low glycerol rapid cooling technique, red blood cells are suspended in 15 to 20% solution of glycerol and cooled at a rate of less than 100 degrees per minute by immersion into minus 197 degree liquid nitrogen, and then stored at below 150 degrees in liquid nitrogen or nitrogen vapor. These will then be rapidly thawed in a 32 to 45 degree water bath and deglycerolized when required for a transfusion. Whereas in the high glycerol slow cooling technique, red blood cells are prepared with a 40% glycerol solution and slowly cooled at a rate of approximately minus 1 degree per minute and stored at minus 80 degrees. Depending on the method used, the glycerol will either be partially removed pre-thaw or all will have to be removed post-thaw. Obviously, if we do have any blood that is available for patients that doesn't require thawing, then that will always be the preferable choice. But this is sort of there as an emergency backup, or in cases where patients are in dire need of blood that we just cannot find a donor for. So I'm going to move on to irradiated components. So irradiation of components is required for patients who are immunocompromised or have other factors that put them at an increased risk of transfusion-associated graft versus host disease. This occurs when donor lymphocytes attack the patient's cells. Irradiation of components damages donor T lymphocytes' DNA, therefore preventing their proliferation and ultimately transfusion-associated graft versus host disease. So to put that in layman's terms, when I was talking about how the red blood cells were separated out, in all circumstances they are leukoreduced, which means that they have a reduction of white blood cells in the unit. Lymphocytes are a type of white blood cells, so the majority of units will not have very much white blood cells in them. However, in cases where patients' immune systems are damaged, even this can be enough to cause issues and cause a type of transfusion reaction known as transfusion-associated graft versus host disease. Therefore, we will irradiate blood units to destroy the T lymphocytes DNA that may be remaining in that unit. It's performed using a combination of gamma and X-rays and is measured in grey. Each irradiated unit must receive approximately 25 Gray to the centre of the unit to be appropriately irradiated. To show that this is effective, each unit has an indicator attached to it before irradiation. This tag should change if the irradiation has been effective. So this is an example of it. If it hasn't worked, then this knot will be visible. If it has worked, then it will be black, and therefore will indicate to not only the person that's performed the irradiation, but also to the lab that it's sent out to and the wards when they are transfusing the unit that the irradiation has been successful. We're going to move on to washed components. So washed components are indicated for patients who have a history of recurrent severe allergic transfusion reactions, amongst a couple of other conditions. Cell washing can be performed by manual or automated methods, but the principle is that the red cells will be centrifuged and approximately 99% of supernatant plasma should be removed and replaced with saline. This is another method that we're using to try and reduce the possibility of having a type of transfusion reaction. We wouldn't use this, say if you had someone come in and their first ever transfusion, they have a reaction to and it looks like an allergic reaction. It would have to be recurrent and we would have to investigate it before we would go down the route of using washed components. But it is an option for patients who do need the transfusions and do meet the criteria for needing components to avoid these type of components to avoid a transfusion reaction. And finally, granulocytes. These aren't routinely used in transfusion anymore, however can be indicated if a neutropenic patient has a life-threatening bacterial or fungal infection. These granulocytes would then be transfused in an attempt to help combat the infection. Granulocytes can only be obtained directly using apheresis. So for anyone who didn't know, granulocytes are a type of white blood cells. They refer to several types of white blood cells, the ones with granules in, which is why they're called granulocytes, because they're very inventive as scientists. It's very rare that these are used. I certainly haven't had to deal with them since being in the lab, although I have seen processes for when they are used. So it's just something to be aware of, that these do exist and were used more so a few years ago. So granulocyte function deteriorates rapidly after collection, therefore it should be transfused as soon as possible after collection, normally within 24 hours. As this product will contain high levels of T lymphocytes, it's normally indicated for immunocompromised patients. Not only that, but granulocyte components are also likely to contain red blood cells, so they must also be ABO compatible with the recipient. And that is pretty much it. These are the main types of blood donations, how they are collected and their uses. You can see that there are a wide variety of components that can be collected from your donation, each of which help patients in different ways. I've gone through some very, very complicated processes and I've only touched the surface, to be honest. I have tried to simplify it as much as possible. But it's important that we're all aware, at the very least, of the basics of how we get these different types of components into the lab and how they get to the patient. And understanding this can really help us in the way that we issue it and to treat the units when they come into the lab. So thank you everyone so much for listening. I hope this has been useful to you. If you do want to read more about this, then please check out the NHSBT's website. They have a lot of resources there. There's even a blood centre tour that you can access on their website, and you can literally, it's free for everyone, and you just go through and click and it will take you to each floor in their blood centre to have a look at, and it will show you all the processes in a lot more detail than what I've gone through here, and probably in a lot more fun because it's got lots of pictures and things for you to click on. Remember to do a reflective learning on this for making your CPD a nice robust piece of CPD, and I look forward to the next talk. Thank you very much. Bye.